In this segment, we will study how to analyze and how to see the expression of the gene in situ. Now, this is the on-site analysis of the genome and how we can perform it. I will illustrate it by taking an example. For example, human chromosome 21 that is involved in the disease known as Down syndrome. And the gene responsible for disorder, that is the 21 gene, is to identify the gene. It would be very useful to know the pattern of expression of this gene or all other genes during the embryonic life of the cell in a human. Now, such studies, they are routinely conducted on lower microorganisms. For example, the mouse genome, it harbors the orthologs, that is the gene in different species that evolved from a common ancestor gene for 161 of the 178 confirmed gene on human chromosome 21. So these are the studies which are routinely performed. Now, the strategy which is applied by the two research groups is, the first research group is the Greger, Ashley, and others, their colleagues. What they did, they looked at expression of 158 of mouse orthologs at 3 times during gestation by in situ hybridization. That is what they did. Now, pattern expression of several genes were found. That is the pattern. And the expression conflict to the specific sites at a specific times. That was also found and studied. Some pattern expression, they were in situ, that is the central nervous system, heart, gastrointestinal tract, and the limbs. And these are consistent with the pathology of Down syndrome, these tissues which they found. Now, the expression of the PEP4 gene in the day 10.5 mouse embryo by in situ hybridization to whole mount section, that is shown in this diagram. And in the day 14.5 embryos, that is by in situ hybridization to embryonic section, is also illustrated in this diagram. Another example which combines the work from Ashley and colleagues and another group which was headed by Ariel Ruiz I. Eltaba. Now, they worked on the expression of the mouse Si3BGR gene in days 9.5, 10.5, and 14.5 of the gestation period. Now, the result of their study, they found that the gene is prominently expressed in the heart at all these stages of the development. As heart is one of the organ which is affected by Down syndrome, the SII3BGR gene is another candidate for involvement in this order. And these guys, they found this discovery or this study has helped us to see that how in situ expression analysis for the gene can be performed.